The word amoeba derives from a Greek word that means change or transformation. The phrase Amoeboid motility describes a mode of cellular locomotion associated with dramatic shape changes. For example, this human neutrophil is an amoeboid cell that changes its shape as it moves. Cells like this explore the space around them by creating highly dynamic membrane protrusions called pseudopods.
If we could zoom in on one of these pseudopods and look beneath the membrane that covers it, we would see a dynamic network of filaments composed of a protein called actin. It is the assembly of this actin filament network that generates force to push against the membrane. Although they look complicated, such force-generating actin networks can self-assemble from just 5 protein building blocks: actin, which forms the structural filaments; profilin, which binds to actin monomers; capping protein, which binds filament ends and terminates their growth; the ARP2/3 complex, which creates new filaments branching from the sides of pre-existing filaments; and finally, a master regulator of network formation called a nucleation promoting factor.
But first, how does a growing actin filament actually generate force? Each filament is like a tiny spring that is continually bent and deflected away from the membrane by random thermal forces. When the filament bends far enough away from the membrane to permit the addition of new subunits, it grows longer. When it bends back towards its equilibrium position, this longer filament pushes against the membrane. This mechanism is called an elastic Brownian ratchet. Elastic because the elastic behavior of the filament is important, Brownian because filaments undergo random or Brownian fluctuations, and ratchet because the continual growth of the filament acts as a one-way ratchet to push forward the membrane.
Branch actin networks grow against the surfaces that are defined by the presence of specific signaling molecules. In many pseudopod membranes, network assembly is triggered and maintained by small G proteins such as RAC and ARF, which recruit and activate a master regulator of actin network assembly called WAVE. The activation of WAVE exposes binding sites for profilin, actin, and the ARP2/3 complex. These binding sites act as conduits through which most of the core components of a branched actin network flow. For example, proline-rich sequences near the C-terminus of WAVE bind to profilin-actin complexes, which are the major source of monomeric actin in the cell. The actin in these ternary complexes can be transferred to the growing ends of nearby filaments. By creating a high local density of profilin-actin complexes, WAVE can dramatically accelerate the growth of actin filaments near a membrane surface.
Profilin-actin complexes bound to WAVE can also transfer actin monomers to a nearby binding site called a wasp homology 2 or WH2 domain. This transfer activity is important because WH2 domains by themselves are not good at capturing actin from profilin-actin complexes in solution. Similar to the actin transfer mediated by the proline-rich region, actin monomers bound to WH2 domains can also be transferred to the growing ends of nearby filaments.
In addition to accelerating filament elongation, WAVE also promotes the formation of brand new filaments branching from the sides of existing filaments. An acidic sequence at the very C-terminus of WAVE binds to an actin nucleation factor called the ARP2/3 complex. When 2 WAVE molecules interact with one ARP2/3 complex and a pre-existing actin filament, they induce a conformational change in the complex and create a new filament sprouting from the side of the pre-existing one. This process of filament creation requires actin monomers bound to WH2 domains of the WAVE molecules. The filament nucleation activity of the ARP2/3 complex is autocatalytic because each new filament provides a new platform on which the ARP2/3 complex can create more filaments.
This positive feedback produces explosive growth in the early stages of branched network formation. As the number of growing filaments increases, however, negative feedback begins to shut down new filament formation so that the network maintains a stable structure. The easiest way to understand negative feedback in a branched actin network is to recall that actin monomers bound to WH2 domains can be used either to elongate filaments or to create new branches via the ARP2/3 complex. This means that growing filament ends actually compete with the ARP2/3 complex for an essential element of the nucleation reaction.
When growing filaments are sparse, most of the WH2-bound actin can be used for nucleation, and the rate of branching is high. As the density of growing filaments increases, however, WH2-bound actin becomes more and more likely to encounter a growing filament end than an ARP2/3 complex, so the rate of branching falls. This negative feedback explains the importance of capping protein in controlling the architecture of a branched actin network.
If individual filaments could continue to grow forever, the negative feedback would cause the rate of filament branching to approach zero. and the actin network would eventually be composed entirely of long, unbranched filaments. By continually removing free barbed ends from competition with the ARP23 complex, capping protein ensures that the rate of branching never approaches zero. In fact, at steady state, the rate of filament branching equals the rate of capping. These simple interactions enable 5 molecular building blocks: actin, profilin, capping protein, the ARP23 complex, and a nucleation promoting factor to self-assemble into a force-generating cytoskeletal motor.
The nucleation promoting factors act as master regulators of network formation for several reasons. First, they control and coordinate the two most fundamental elements of network formation: the creation of actin filaments by the ARP23 complex and the elongation of filaments by the addition of new subunits. The coordination of these two processes is possible only when nucleation promoting factors are clustered on a membrane surface. The localization of nucleation promoting factors to membrane surfaces also gives polarity to the actin network, enabling it to push against the membrane.
Of course, after they are assembled, branched actin networks must also be disassembled.
But that's a story for another time.